All right, hello biology people. Hope you're doing well. Let's get today into our discussion. We continue with chapter 3. So up to this point, you should now know how macromolecules are formed by the process of dehydration synthesis and how macromolecules would be sort of split apart, destroyed through the process of hydrolysis. So there's 4 macromolecule categories. Today we begin our discussion on the first, which are the proteins. Again, I'm kind of certain that most of you have heard of what a protein is. You have some idea of what a protein is. A protein is going to be sort of derived from sort of these types of food products, chicken, fish. I don't want to get into all the nutritional stuff just yet, but what a protein is, it's a combination, it's a polymer, it's this big macromolecule made up of smaller amino acids. So a protein is a large aggregation of amino acids. We're going to talk about what amino acids are right now. So there's going to be different families of proteins. These different families are going to have different amino acid compositions. The different families of proteins will have different shapes, and they're going to have different shapes which allow them to undergo different functions in the body. So we're going to have some supportive type proteins that support the body, a lot of connective tissue, things like ligaments, tendons, skin, a lot of proteins that help make our body shape the way it is. Transport proteins, which things like hemoglobin that transport oxygen throughout the body. Movement proteins like muscles that can contract and stretch and relax, generating force, that kind of thing. A lot of protective proteins. You might have known about our antibodies, immunoglobulins. These are important proteins that circulate in our blood and help fight off disease, things that we've encountered in the past, things that we're newly sort of being subjected to and trying to build up immunity there, right? And we've hinted on these a little bit, the idea of enzymes. So we have a whole family of proteins that function as little dehydration synthesis enzymes that are putting things together, and other families of proteins that act as hydrolytic enzymes that are breaking things apart. So very diverse group of macromolecules, a lot of different functions, these proteins, right? So very important for normal biological health, biological physiology. So fibrin, blood clots are proteins. Feathers, webs, human skin, human nails, human hair, all of these comprise of proteins, right? Different families of proteins. So before I continue with proteins, I got to take a step back and we got to look at the individual building blocks of a protein. So proteins are polymers of amino acids. They're all this aggregation of little units that we call amino acids. So amino acids are the monomers for proteins. So it means the same thing, just phrased differently. So proteins are polymers of amino acids. Amino acids are the monomers that make up the proteins. And what you're looking at here, we have 3 examples of amino acids. And I'm not expecting you to be chemistry experts at this point. I just want us to start sort of identifying some important parts. So I want you to see this part here. If you're keeping up and you remember your functional groups, you know that this is an NH2. This is an amino group. And here we have a hydroxyl, a carbonyl, collectively we know as a carboxyl. So and we have this white rectangle with some other unique feature there. But the basic framework, the basic sort of molecular skeleton we see here. We have an amino on one side and a carboxyl on the other side. Some other mess here, right? Amino on one side, carboxyl on the other, some other mess here. Amino on one side, carboxyl on the other, some other mess there. So this is the idea for amino acids, right? I want you to get that idea. Amino on one side and carboxyl on the other. So we know that proteins are going to be formed by these amino acids combining together. And you already know the mechanism of how this works, right? We have these enzymes that come in and instigate this. But if I'm looking at one amino acid, that little circle with the R represents this sort of, it's like a variable, whatever's unique to each individual amino acid. So phenylalanine is different than tryptophan, is different than tyrosine because of what's in this little white rectangle there. So this is a unique composition for that particular amino acid, but we know that each amino acid will have an amino group in yellow here and a carboxyl group here. An amino and a carboxyl here. Well, what happens? So the hydroxyl from one carboxyl will be removed, and the hydrogen from the amino group will be removed to form water, thereby allowing the carbonyl, the carbon of the carbonyl, to connect with the neighboring nitrogen of the amino. And again, that is where that bond will form. It's a covalent bond. We call it a covalent bond. Biologists call it a covalent bond because that's what it is. But chemists like to get real specific, and they call these peptide bonds. A bond, a covalent bond between two amino acids. So it's the basis of a protein. So again, once we connect two amino acids together, we don't call them two amino acids, we call that now a protein. This is the basis of building a protein. I'm not going to have you memorize these. You'll have to do this later in your biochemistry class, but for now, I just want us to see, well, there's different sort of structure, different behaviors, but there's different sort of types of amino acids, but they all have that similarity, right? Amino, carboxyl, amino, carboxyl, amino, carboxyl, amino, carboxyl. They differ in their variable region there, but that bottom part is unique for all amino acids. So if I backtrack, this dehydration synthesis event, it can function with any combination of amino acids because they have the ingredients, they have a sort of a hydroxyl that we can work with, an amino and their hydrogen that we can work with to undergo dehydration synthesis. So all of these can be mixed and matched in all kinds of different ways. Now, there's roughly 20 amino acids that exist. The body needs them, and we need them every day. But our body has figured out that, man, these humans don't eat very healthy. They don't eat a nice array of foods. So what your body has done, it's kind of worked the deal with you. So you may not be able to eat every single one of these each day, but as long as you eat these 8 essential amino acids, right? These are the bare minimum that we have to ingest on a daily basis. So if we get these 8, the body can restructure and reconfigure them to get all of the ones that we need. But these are the 8 essential amino acids that our body can't make. We have to ingest these, and then once we have these ingredients, then we can build other amino acids and the corresponding proteins. So I don't know if you eat a variety of foods. I don't know if you supplement. If you don't eat healthy, you may want to consider your little SpongeBob vitamins or some kind of multivitamin supplement. But yeah, you need these. And how many do you need? It depends. How active are you? Are you training for a marathon? Then you're going to need these in a high amount. Maybe you sleep all day. I don't know. Then you're going to need these, but not in as much abundance here, right? But realistically, if you eat a varied diet, you probably get these in your diet on a regular basis, right? On a daily basis. So you can think of here in El Paso, a common dish in the Hispanic community would be some dish with bean products and some dish with corn products, right? Corn tortilla and beans, a little taquito of beans, something like that. It's not a glamorous meal, but the beans would provide then the 6 of these essential amino acids, but they're deficient in 2. The corn product would provide 6, but it's deficient in these 2. But by corresponding these 2 types of foods, beans provide what the corn does not, corn provides what the beans do not, and that gives you that full array of amino acids, right? Not a glamorous meal, but how many times did grandma and grandpa eat, little taquitos of beans, right? Corn tortilla and beans, right? They probably eat these a long time, and they made it to be old, and they're hopefully healthy, right? So the idea of a balanced diet there. So once we have the amino acids that we need, the body can then start to manufacture proteins. And it's going to do this sort of in a structural level. So when I talk about structural level, it means something very specific for proteins. It's the sequence in which the protein is being built. Proteins will progress through 4 structural levels, right? Primary first, secondary second, tertiary third, and quaternary fourth. It would be easy to say first, second, third, fourth, but that doesn't sound intellectual enough. So these biochemists term it as primary, secondary, tertiary, and quaternary. The primary structure is kind of simple. This is that reaction, the dehydration synthesis reaction that connects the amino acids together. So they make our chain of amino acids. That's the primary structure, the precise sequence of amino acids. And I like to think of primary structure like these little children graduating from kindergarten. I don't know if you remember that day. You kind of graduated kindergarten. Yeah, got my diploma. Let's say little Susie wants to be a doctor when she grows up. Well, she graduated. She's got her diploma from kindergarten. Is she ready to go into the surgical room? I don't think so, right? So yes, this is a protein. It's a primary structure for protein, but it's not yet ready. It's not complex enough. It needs more training, more chemical sort of structuring. But yes, it is a protein. Yes, she did graduate, but needs more time, more knowledge, right? Yes, it is a protein, but it needs more complexity at this point. So we continue. And now we get into the idea of our dehydration synthesis again. So connecting one amino acid to a second amino acid, generating our water, and then our peptide bond, our covalent bond. What this picture is trying to represent, these little pink or purple circles, are trying to represent individual amino acids. So these that have bonded together by dehydration synthesis to make this long chain of amino acids. So the primary structure of the protein. So we have this long chain, and then because of hydrogen bonding, because we have delta positives over here, delta negatives over here, that primary sequence starts to either spiral, can't really do a spiral too well, but a spiral or a pleated sheet, kind of up and down, up and down, up and down. So these are 2 common structures of the secondary structure. So again, they start to spiral, the alpha helix, or up and down the pleated sheet like an accordion. So these are products of the hydrogen bonding, delta positive and delta negative there. So we have our long chain of primary structure, and then we have little sections where we get into our pleated sheets and our alpha helices there. Now, this is like graduating from middle school. Maybe you graduated from middle school in 5th grade and you think you're all cool, right? Before you progress onwards, or you graduated from elementary, or I don't know when you graduated from, but you're still not quite ready. You're still not ready to be a doctor. Little Susie's still not qualified yet to be a surgeon. So she must progress to the next level. So here we get to the tertiary level. Tertiary level, this is when the shape of the protein is finally completed. That protein is fully functional at this point. So we have to, ding, 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 test question here, right? So we have to get to the tertiary level in order to have a functional protein. This is when they're fully functional, right? They're mature, quote unquote. So that little sort of purplish chain is represented by this little purplish ribbon. So amino acid backbone. But then we start to stabilize and we make it into a three-dimensional shape. Now it's fully functional. And tertiary structure reminds me of a high school graduation. Maybe some of your early college students, I'm not sure, but if you remember graduating from high school, let's say you're 18, you graduated from high school, like, yes, I graduated from high school, I'm an adult, I'm on my own, I can do whatever I want. And then, oh, damn, I'm an adult. Now you gotta kind of go to work. Maybe you gotta, you go to the military, you can go to the workforce, you can stay home and be a bum, you can go to college, whatever the case. But you're kind of functional as an adult, and society views you as a legal adult at that point as well. So the idea of a completed protein, tertiary structure. So we go from the primary, secondary, and then to that three-dimensional mess of proteins here. And for a lot of proteins, it ends there, right? It's the end of the line, right? End of the story. For some proteins continue to get more complicated, right? Just like some of your friends graduated high school, that's it. But you graduated high school and you come to college to get your brain more complex, yeah? So the quaternary level, quaternary structure, results from the aggregation of multiple tertiary proteins. So see if this makes sense. So you have a tertiary protein that can do its job, it's functional, but it may decide to team up with another tertiary protein, and that then we would term as a quaternary aggregation. It's not always necessary. I don't know how you study. Maybe some of you study at home by yourself, but maybe some of you collaborate and you form like study groups. Well, that would kind of be like a quaternary structure. You're functional and your buddy's functional, and now you start to collaborate, and that makes like a team, right? The quaternary structure. Hemoglobin, the protein that forms hemoglobin, functions in this way, right? So there's one functional tertiary, another functional tertiary, another functional tertiary, and another functional tertiary that are all interacting together to form then a functional quaternary protein. So they don't have to do that. Each one could function on its own, but it elects to work as a team. So again, summarizing: primary, secondary embedded within, and then finally we achieve our functional tertiary. We can stop there, be perfectly fine, or we can team up with others in our quaternary structure. So again, you can see a quaternary structure. There's a blue functional protein, a green functional protein, and there's these aggregations that all collectively work together. So not too bad, I don't think, right? Protein structure is not that terribly complicated. Just understand we got to get at least to here, and we can elect then to get to here or not. Right? The quaternary is optional. And I'm going to pause this here. I have one more little aspect to cover for chapter 3, and that will assign then another little homework on Pearson and then on Labster as well. But not too much stuff to soak in today there.